Hello everyone. So I'm the last speaker from the Mekong Livestock Research Team today, and I'll be focusing on reproductive pathogens in large ruminants in Laos with a focus on neosporosis, brucellosis, leptospirosis, and BVDV. And the analysis in this investigation was conducted and facilitated by the College of Veterinary Medicine and Life Sciences at the City University of Hong Kong. So just to rehash why it is important that we develop the efficiency of agricultural production in Laos. Firstly, it has an important role in food security, which is needed to accommodate for the population in Laos, which is expected to grow by 42% by 2050. And second of all, to improve income and livelihoods because cattle prices currently reside at an all-time high. So the problem is that reproductive efficiency is challenging this because it is constrained. So in addition to the 9-month gestation period in cattle, on average there is a 5 to 7 month anestrus period. And for buffalo, in addition to the 10.5 month gestation period, there is almost a 10.5 month anestrus period. So the reason why we have inefficient reproduction is predominantly because of a lack of nutrition, because no nutritional inputs are put in by farmers who mainly focus on rice production and cash crops. But there are also diseases that can potentially cause abortion, and we have farmers anecdotally reporting that they do experience abortion, and this will threaten efforts to improve the efficiency of agriculture in Laos. So of the many potential threats, disease threats causing reproductive loss, we focused on brucellosis, lepto, BVDV and neospora, on the basis that we have evidence of infection and reproductive loss in neighbouring countries. Laos is increasingly engaging in trade with these countries, and we suspect that farmers are engaging in risk factors at the village level. So I won't go into transmission, but just making the point that all of these pathogens can cause abortion. Their transmission is possible in the village context, such, for example, for neospora, we have the presence of semi-domesticated free-roaming dogs, and we have warm, humid conditions in the wet season. And of particular importance, leptospirosis and brucella have zoonotic potential. So for this investigation, we were very lucky that we were given access to 160 samples from the serum bank at the National Animal Health Lab in Laos for the preliminary study. Based on the positive findings, we were then given access to more samples in 2018. And they were newly collected samples of 520. We did a proportional randomized selection at the province level based on data that we had that generally there is significant provincial differences. We tried to keep the sites consistent between the two studies, but we didn't have any samples from Savannah-Ket province in 2018. We used commercially available antibody ELISA kits, and the analysis occurred at the City University of Hong Kong at their veterinary diagnostic lab. So the results showed that we had really high neospora in buffalo of approximately 70%. This was significantly higher than co-reared cattle of 8%. We had low BVDV in both cattle and buffalo, 5 and 10%, which were not significantly different. For leptospirosis, we had approximately 20% seroprevalence in cattle, which was significantly higher than co-reared buffalo of 3%. In our 2018 study, we basically had the same results, with the exception that neospora was slightly higher in buffalo of 80%, leptospirosis a bit lower in cattle of around 13%. So the key points from this is that the study allowed us to show that there is evidence of reproductive pathogens in Laos. And this provides the justification that is really critical to progress investigations into production impacts. And it also shows that there's a really strong need to develop abortion surveillance so we can understand how big this issue is. Because farmers don't keep records, it's something that we need to put more emphasis on. So because we have these significant differences between species, it also suggests that maybe that we do have a maintenance species. Potentially there is a risk to co-rearing the two species. And lastly, the species difference also suggests that maybe we have farmer practices conducive to increased risk. An example of this is the superior care of buffalo in certain areas, because they were traditionally used for draft power and they are a higher value animal, they may be kept closer to the home, closer access to dogs, whereas cattle might common graze the roadside for weeks. So we wanted to see where the infection was, and we found that for neospora in buffalo on the left and then in cattle on the right, we basically had the highest seroprevalence localized in Luang Prabang province, which is the tourist capital, and we predict that this has a higher meat consumption and therefore higher domestic trade of animals towards this area. In the 2016 pilot, the same trend was also reflected, except we did have buffalo samples from Vientiane, which is the capital province, where we do have a lot of animals being harboured, and we think it just confirms this link. So here we have BVDV in cattle on the left, lepto in cattle on the right. Again, we see the same trend of increased seroprevalence in either Luang Prabang or Vientiane. And I just want to point out that for lepto in cattle, we've got 30% seroprevalence in Vientiane province compared to 7 to 13% in the remaining provinces. The same trend was observed into the 2016 pilot study. So the ununiform seroprevalence across the provinces and significant differences suggests that there are provincial level risk factors. We suggest animal transit as a potential reason. And what this map just shows you is that Laos is like a thoroughfare for animal trade in Southeast Asia. You've got trade from Thailand going south across to Vietnam, trade from Thailand and other parts of Southeast Asia north to China. And these are just the official movement pathways, and there are many, many unofficial ones. So this is maybe something that is facilitating the spread of disease. And of course, we predict that there are maybe farmer practices also conducive. So we also found that neospora in buffalo generally increased with age. So we used the Fisher's test to confirm this, but we also used a logistic regression where we were able to accommodate or account for the background noise by including province, district, village, and the farmer as random terms, again showing that age was significant. And when we predicted the probabilities, this graph shows that when buffalo are born, there's a 60% chance that they are seropositive, suggesting that there is a vertical transmission, which is quite efficient. But because this increases over time, it also implicates village dogs in the transmission. We also found that lepto was significantly higher in the wet season compared to the dry season, suggesting that the warmer, moist conditions favours leptospiro survival. And we thought this was quite important because in one of our villages in Vientiane, we had 40% village prevalence, which was, much higher than the overall prevalence. And what was concerning is that this village is located only 1.5 kilometres from a pretty big water-based tourist attraction. So not only do these pathogens affect productivity, but also tourism and human health. So we opportunistically followed this study up with a bit of a risk factor survey where we revisited 75 farmers involved in the blood collection. We went to all the provinces and we randomly selected 2 villages per province. So we conducted a survey assessing risky practices, keeping in mind that there was a gap between blood collection and the survey, and we spent 2 weeks driving all around Laos to conduct the surveys. So from the survey, it confirmed that we have farmers participating in the known risk factors. So for neospora, 60% of farmers reported that their dogs were consuming regularly aborted fetuses, placentas and dead calves. 16% of farmers reporting that dogs and rodents were defecating near cattle feed or water. 40% of farmers reporting that they didn't remove any feces from calving areas. And for BVDV, 16% of farmers had introduced animals, including pregnant animals, within the last 2 years. 20% reported that their cows shared bulls. 75% were engaging in common grazing. And for lepto, we had 40% of farmers allowing animals to graze near flooded rice plots, and 20% had experienced severe flooding in the last 5 years. And we were also able to gauge the rate of reproductive issues, which there was no data on previously, and about 20% of farmers reported that they experienced reproductive issues. And we also assessed knowledge and basically found 40% of farmers were aware that large ruminants abortion could be caused by disease, and then only 36% of farmers thought that their family could become infected from diseases from cattle, and 24% of farmers thought that large ruminants could get diseases from dogs or rodents. So I think what this suggests is that farmers are engaging in risky practices because they don't realise that they can cause diseases in cattle and humans. And by addressing knowledge, this is potentially how we're going to start changing those practices. So just to summarise, it's more than likely that reproductive diseases are going to become increasingly important in the future. We're starting to have increasing herd sizes, we are getting increasing productivity, and there will be increased transboundary trade. So I just want to touch upon my colleagues' work. So not only reproductive diseases, but of course foot and mouth disease, hemorrhagic septicemia and other transboundary animal diseases, as well as internal parasites, are going to continue to threaten efforts to enhance reproductive efficiency through nutrition, animal health and breeding management. And it's extremely important that we investigate these diseases and prevent disease. Thank you.